Our project is the development of a vaccine for contagious bovine pleuropneumonia. Involved originally 3 primary organizations, VIDO, Intervac, Calro, and ILRI from Nairobi, but a variety of others as well. The Department of Veterinary Services, for example, has played a large role in a variety of other organizations within the country. So what is the disease? It's caused by a bacteria. So you heard about viruses. Now we're talking about free-living organisms. And this is Mycoplasma mycoides. It's capable of causing acute or chronic disease and kills somewhere between 15 and 30% of animals that it infects. It's transmitted very, very easily by aerosol. And we think that natural immunity, you require both so-called cellular immunity as well as humoral immunity. So for the plant people, essentially antibodies on the humoral side as well as cells, specific cell types on the cellular side. And it's important throughout Africa, specifically sub-Saharan Africa. Economic cost, it's interesting. If you look at the literature, it's anywhere from 60 million to 300 million. You'll find different estimates. So this is the lower end of the scale. I think it's safe to say, though, it does cost significantly more than that. Through mortality, reduced meat and or milk production, and it remains a threat to Southern Europe because there is the movement of animals and animal products, albeit below the table from Africa into Europe. And in fact, there was a case in I think the last case was the late 1990s, 1998, of CVPP in Portugal. And that was due to an animal moving from Northern Africa across over to Portugal. Control has historically been via vaccination as well as movement control. We used to have the disease in North America many years ago, Australia, Europe, etc. And traditional vaccines as well as movement control, culling of animals, etc., managed to eradicate it. When we look at things like movement control of animals and culling of animals, first of all, culling is something socially that is coming under a lot of scrutiny. If you look at foot and mouth disease in the UK, for example, the photos of pits of animals that have been killed, the public simply isn't accepting that anymore. And the movement of animals in an African context is far more difficult than you would see in Europe or in North America. Antimicrobial usage is an option. This is a bacteria. You can kill it with antibiotics. 2 points. First of all, it is expensive, up to $18 per animal. And I would argue it's not sustainable. If you look at the issue of antimicrobial resistance throughout the world today, and particularly in the livestock sector, this is a major driving force in the vaccine field, in the animal disease field as we move forward. And as Lauren mentioned, the linkages between human and animal health is the major driving factor in that area. The vaccines that are used, live vaccines, have historically been the best, and these are attenuated bacteria. Again, Lauren mentioned passage in eggs or broth in this case, and the results are mixed. The vaccines tend to work, but there's a couple of problems with them. Again, Lauren mentioned thermostability. A live organism is killed easily by heat. So you need refrigeration. You need a cold chain from production all the way through to administration. The other thing is they tend to cause fairly severe tissue reactions when you inject the bacteria. And I can tell you in the Canadian context what that would cost. That would be about $9 that you would pay per animal due to those tissue reactions. Killed products do exist. Protection is not as good, and I would argue they simply don't work. And you need to include immunostimulants or adjuvants with them. And finally, the subunit vaccines that have been under development for the last decade and a half, very little protection has been observed, and in some cases there has been exacerbation of disease, and that's simply because we don't know what the proteins do. So the goal that we had, and this was during phase 1 of the CIFSRF program, was to identify protective antigens from Mycoplasma mycoides, using a reverse vaccinology approach. And I'll tell you what that is in a second. For production of a subunit vaccine. So this is not a live organism. This is purified proteins that we are talking about here. So it's not a GMO. It is the product of a GMO. And finally, or secondly, demonstrate vaccine efficacy within a 30-month time frame. Now, I cannot tell you how difficult a target that is. In 30 months, what we had to do with our Kenyan colleagues is set up a collaboration across an ocean. And we had to get the technology, the animal models, et cetera, going within that window. This is virtually unheard of in the vaccine field. And this is the power of the technology such as reverse vaccinology. So what is it? So this shows traditional vaccine development. Normally, we start out with a thorough analysis of the biology of an organism. So we understand pathogenesis, the function of proteins, et cetera, and we move over towards vaccine development. So, for example, we know with cholera. Cholera produces a toxin that is important for disease and important for immunity. So one focuses on that as one way. Likewise with pertussis, we know the virulence factors that are involved in disease. So for the acellular vaccine, there's a total of, I think it's five, Volker? Yeah, 5 components in the vaccine. So it's really biology leads to the vaccine. Reverse vaccinology is exactly the opposite. What we do is go with no knowledge of the function. All we need is a DNA sequence over here. And we simply identify, using computational methods, every potential vaccine antigen or surface component on the organism. We test them, and that leads back into a study of function once you've identified what might or might not be protective. We do have one example of a vaccine developed that way that is on the market right now. That was done by Novartis. This is a human vaccine. And what they were looking at Neisseria meningitidis, serotype B, for which there was no vaccine. And this shows the pathway they followed. So computational prediction of vaccine candidates. They had 600 of them. They expressed 350 in E. coli. They did mouse immunization experiments, identified 91 novel surface-exposed proteins. 28 had bactericidal or were able to induce bactericidal activity. And ultimately, they ended up with a vaccine that is actually on the market today. It took a long time. This was a human vaccine, and this was the first example of this type of technology being used, but a very, very powerful technology. Advantages of reverse vaccinology is speed. We can identify all the antigens we want to test within a week. The computers do the work. Now, obviously, computers are programmed by humans, so there is a factor in there that unfortunately we still control, but it is very, very rapid. It removes human bias from the process of vaccine testing, and I cannot stress how important this is. When we look at lists of gene products, and we being the microbiologists, molecular biologists, and vaccinologists, we always focus in on what we're familiar with, and that is not a good idea. Not for organisms, complex organisms, like Mycoplasma or indeed like Neisseria meningitidis. We don't need to be able to grow the organism. Now that has a couple of advantages. First of all, if it's a level 3 or level 4 pathogen and you don't have access to facilities, well, it doesn't matter. All you need is DNA to move forward. Other organisms, there are a number of them, especially aquatic pathogens, where you simply cannot grow them, but you do have a genome sequence and you can move forward that way. And all it relies on is the availability of robust animal models. In the human vaccine field, this can be problematic as they rely on mouse models. And to quote Lauren, mice lie. We rely on natural animal models. They're not really animal models. They're in fact real disease. So here's the approach we used with Mycoplasma. So again, screen computationally, We had predicted we would come up with about 200 targets, clone and express in E. coli, rank for vaccine testing, and the ranking was simply done by a series of methods, including do they induce immune responses during a natural infection, that type of thing. And then test pools of proteins for protection, ultimately end up with 2 to 4 individual proteins that we could use in a vaccine. So this shows the procedure we use for antigen selection. And again, I'm not going to go through this slide, and I don't expect you to. All I would point out is we started off with 985 different potential antigens that might be useful. The final list we came up with was 82. So the computer modeling was able to get that list refined over 10 times to what had to be tested. Again, trying to do that biologically, there is no way you could do that even given years. This is a list of the proteins. Again, don't expect you to look at it, but I show this to raise an important point. So out of the list of all of these, 8 of them have a known function. You can see them here, 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 and a couple over here. There's a variety that are listed by their structure, i.e. they're a lipoprotein or something else. But there's a large number that, in fact, are hypothetical. These are things that you don't study if you're looking at biology. Okay? You simply don't know what these are. In fact, you don't even know that they exist, looking at conventional approaches to vaccine development. Whereas here, probably about half of our list is composed of unknowns, totally unknown. So for antigen testing, the way we did it was to pool antigens in groups of 5, combined with an adjuvant. We gave 2 immunizations. And when I say we, this work was all done in Kenya by our colleagues at Calro and ILRI. We challenged with mycoplasma, then ultimately rescreen any antigens that appear to be protective, and then look at a final vaccine formulation that will address things like thermostability, et cetera. And what we are hopeful at the end of this is we will get a, I wouldn't say a single shot, but something that requires maybe 2 immunizations to start and then an annual immunization or boost. The animal model used, we used Boran cattle in the initial studies. It's an intratracheal challenge. Again, we vaccinate, boost, challenge, and then daily clinicals and post-mortem analysis after infection. This shows a summary of some of the results in one trial that was done. So here you can see this group N, for example, in terms of lesion scores in the lung and pathology indexes, was significantly better than any of the other groups, including the control. And interestingly, when you look at what was in that group, there are only 4 proteins in this. They're all hypothetical. We have no idea what these proteins are, other than they protect against disease. Results of another set of antigens that were tested. Here you can see that there's a couple of groups that actually look better. Group C, in fact, looks like we got sterile immunity. We could not culture the organism from the lung, although I'm sure it was there, in that there was some pathology and certainly some lung lesions. And again, what we're looking at here is 2 hypotheticals, 2 lipoproteins, and 1 protein of known function, elongation factor 2. And interestingly, this particular one is not something that you would intuitively pick. And I apologize for my voice. I managed to damage my vocal cords and never know how long they're going to last. Elongation factor 2 is interesting because it's an intracellular protein. It's not something on the outside of the cell, so it's not something that vaccinologists would traditionally pursue. But again, the computer was able to pick it up, and in fact, I always say, Lauren, you should remember, but I think both of us will probably forget, back in the 1980s, we actually identified this protein at VIDO here in another respiratory disease of cattle caused by, then it was called Pasteurella hemolytica. And we threw it out because essentially everyone said, it's intracellular, it's not going to work. Well, it does. And in fact, we know it works with a variety of other species as well. So that was the end of phase 1. So within that 30 months, we had prototype vaccines, an incredible accomplishment. And I would argue that that technological side, the identification, cloning, et cetera, was relatively easy. Again, our Kenyan colleagues had to do the bulk of the work with the animal experiments. These are not easy to do. Phase 2 of CIFSRF, what we are looking at is defining or refining the vaccine formulation, including the choice of adjuvant, route of delivery, dose and duration of immunity. And again, these are things where we have tried to incorporate feedback from the socioeconomic team within the project as to what do people want. You know, we're not going to try and dictate that this is what the vaccine is going to be. Use it the way we say. We want that information, as Lawrence said, in order to ensure that it is adopted. Whoops. Scale up production of vaccine components from recombinant E. coli. That's being done with Kevivapi. Defining the regulatory pathway in Kenya and elsewhere. This has been an interesting process in that there isn't a robust regulatory framework that we can follow with this type of product. We did have a workshop, I'm trying to remember when it was, I believe it was last December in Nairobi, and I think it was a very encouraging workshop in that we were able to get all of the stakeholders in the same room for a day and sit and work out some of the issues that we face in defining how this should move forward. Again, it's not a GMO, it's a product of a GMO, so there are some issues that we need to address. But I think we know what they are, and we will be moving forward. And finally, testing the vaccine under field conditions. Everything we've done to date has been essentially under controlled experimental conditions at the Calro and Ilri facilities. This needs to be taken out into the real world to ensure that it performs exactly the same. And again, mentioned the socioeconomic group. We're going to hear, I believe Salome is going to be talking this afternoon as part of the panel on some of the issues that we faced and the studies that have been done, so I will not mention that at all. So again, these are the targets that we're looking at for phase 2 that were identified in phase 1. We eliminated 1 protein, so we've got a total of 8. 8 proteins. To make that economically is virtually impossible within the context of a subunit vaccine. So what we've done is come up with 2 different approaches to do this. One of them is we actually have made chimeric antigens, and you can see here, so here's an antigen by itself. You combine them together, you get a higher molecular weight protein, and you can then get away with using 3 or 4 different components in the vaccine. Is that economical? And the answer to that is yes. We know that simply because we've done it before, and other companies have done it before as well with this type of recombinant protein. In terms of issues as we move forward, manufacturing clearly is one. Manufacturing of recombinant proteins is different than what one faces with traditional vaccines, traditional bacterial vaccines, in that traditional vaccines, you're looking at biomass. So accumulate as much biomass as possible. This is a product. Biomass doesn't matter. It's how does one isolate that product out of it. So it requires a conceptual change in mindset, and as well different equipment and so on as one moves forward. Regulatory, I mentioned, is certainly a challenge in that our Kenyan colleagues are essentially helping write the rules as we move along for this type of product. And vaccine rollout. Is this going to be a vaccine that ultimately is used for eradication, or is it going to be used for disease control? It makes a big difference in how one defines the trials that need to be done and how one rolls the vaccine out. I would argue eradication in the case of a bacterial disease is virtually impossible. These are free-living organisms that they don't require an animal host to survive, and trying to eradicate that type of disease is virtually impossible, whether it's in a human population or indeed whether it's in an animal population. Having said that, disease control becomes relatively easy, which functionally becomes the same as eradication. What are the benefits, the impact? The benefit of farmers, up to $60 million annually. This graph here shows the return on investment for a typical recombinant vaccine in the animal world. And I think the numbers don't matter, but to point out that here's the investment, here's the return, and this, in fact, these are figures out of Saskatchewan. It's about a 20 to 25 to 1 payoff. It doesn't get much better than that as an investment, and that's what it's all about. It is an investment, it's not a cost. Capacity building. We have an opportunity to go to that next level with manufacturing. These are manufacturing processes and facilities that today don't exist in Canada. They used to, but they don't anymore. Here's an opportunity to do something in Kenya and elsewhere in Africa. And finally, international trade and movement of animals and their products. Ultimately, what we would like to see, of course, is the ability to have animals legally move within the continent, but as well outside of Africa into the European community and elsewhere. And finally, it's a platform for other vaccines. So contagious caprine pleuropneumonia in goats, we should be able to plug that right into what we've done with CBPP. I'm not saying you're going to have the same antigens that come in the vaccine, but we have the pathway there. And indeed, I would argue that it's not going to take 30 months to do that. In fact, probably you could do it in about a year and a half. So, to summarize, reverse vaccinology has provided a valuable tool in our view for the development of a vaccine for CBPP. And in fact, we're currently using that for a variety of other diseases, including Johne's disease, bovine tuberculosis, as well as CCPP at VIDO Intervac. Thank you.
And I think, you know, that was also a very good example of what we were talking about earlier on, that it is possible to do discovery science and mobilize it directly to where the biggest need really is. Any questions for Andy? Yes.
Yeah, okay, thank you very much. I really appreciate your presentation. Even I decide like to take your field of study. You presented very nicely. I mean, I'm from Ethiopia. East African countries are really suitable for vaccine production because the air condition is very nice, and it's also a lot of assessment also done regarding the vaccine production for human and also animal. But in your presentation, what is the possibility of doing the level of technology to be done in Kenya or Africa? Because we need the human resource, we need the level of technology, we need the network, we need a lot of animals also are living in African countries. I mean, what is the difficult to make the level of technology to make the business in whether it is can be in East African countries, because it's very important. Otherwise, doing the research activities, biosafety aspects, and so on, cannot be enough, in my opinion. That's why I think we have to, if it is possibility, the possibility to have do some aspects. If you go to Ethiopia, for example, we have one vaccine producing industry. It is 50 years old. They are producing different 19 types of vaccines and distributing to some African countries. But the level of technologies, especially the bioreactors and so on, are obsolete. Then really, are you really committed to make the level of technology to produced in Africa?
Well, the short answer is yes. And I would argue that the level of technology required to produce this type of vaccine is actually much simpler than that required to produce conventional vaccines. And indeed, the first recombinant vaccine for cattle was developed at VIDO many years ago. And in 1991, I believe, the production of that vaccine for the Canadian market took place in the VIDO facility in a room that was the size of this little stage here, with equipment that virtually anybody had. There was no fancy new equipment required. It is simple technology, very, very simple. But it is not necessarily compatible with the conventional way of doing it. So you have to look at things a little bit differently. Again, it's not simply growing organisms, killing them, mixing them up and vaccinating. That what one has to do is set up methods for purification or enrichment that are not difficult. Again, very, very simple to do. But again, that different mindset. You have to focus on that final product as opposed to the bacteria that you're using to grow it. So I would argue that this is pretty simple stuff. Will it require an investment? Yes, it will. It's going to require an investment in terms of there is some equipment that is going to be needed, but as well in the human capital side. But I think that's achievable, and it's a way of leapfrogging to that next generation. I mentioned Canada doesn't have this capacity at the current time, largely because of the economic downturn in 2008. I think this is an opportunity for Africa to actually move ahead. Not only with this vaccine, I think the vaccines Lauren described as well.